Thank you, Song. I'm very honored to introduce the next speaker, Dr. Martyn David Kirk. Professor Kirk now is the National Health and Medical Research Council and the Career Development Fellow, Australian National University. Dr. Kirk has been in epidemiology and worked as NHMRAC Career Development Fellow at the Australian National University. Previously, Professor Kirk has run the Australian Field Epidemiology Training Program and coordinate foodborne disease surveillance for Australia. Professor Kirk is the acting chair of the TEPHINET Advisory Board and a member of the Strategic Leadership Group for the FETP enterprise. Professor Kirk is a member of the WHO Advisory Body, the Foodborne Disease Burden Epidemiology Reference Group. And today, his talk is a recipe for good investigation of foodborne disease outbreaks. Let's welcome Professor Kirk.
Thank you very much, Dr. Cheng. So it's a great privilege to be here today. I have been to many TEPHINET conferences, and I'm so disappointed that I can't be in Taiwan with everybody today, and we could go out to the night markets and have lovely food. So today I'm going to be presenting the do's and don'ts of foodborne outbreaks. And I want to take the approach of a recipe book. I love cooking, I love food. And so I think there's a lot we can learn from that, and it serves the analogy well. Before I start, I'd just like to really thank the organising committee for inviting me. I particularly like to thank the people who have organised this committee. So SafetyNet have done an amazing job, as have the Taiwan FETP and the Taiwan CDC. So, and I also would like to pay my respects to the traditional owners of the land on where I'm meeting today, which is the Ngunnawal people. They're the local Aboriginal tribe that has an ongoing connection with the land here, and I pay my respects to their elders past, present and future. So next slide, please. So I'm just going to talk a little bit about why do we investigate outbreaks in the first place, and then I'm going to give you a basic recipe for investigating outbreaks. I want this to be an opportunity where we can think a bit more deeply about how we investigate foodborne disease outbreaks. And you'll see here I've got one of my favourite cookbooks, which is the Hanoi street food, and I love street food from Vietnam and from many other countries in the region. I'm going to give a bit about the context for the recipe that we might use to investigate an outbreak, and then talk about what are the key ingredients. So looking at exactly what kind of things do we need to have in an investigation that will give us the answer we want. Then I'll talk a little bit about methods we should use. And then finally, I'm going to come up with a list of do's and don'ts and make some general conclusions. Next. So why do we investigate outbreaks anyway? Apart from them being a great training opportunity for FETP fellows, there's a range of other reasons, and these are most important. Firstly, we want to understand the source of illness. We want to try and identify the specific food and even the specific ingredient that is causing the outbreak. The outbreak may be ongoing. Cases may still be occurring. Not all the food may be consumed, and particularly if it's a commercial food product. I'm going to talk a little bit today about an example where there was a commercial food product, and you can see what the consequences are. Another reason to investigate foodborne disease outbreaks is that they're a sign of a food safety failure. And we want to understand what those food safety failures are, because they help us identify how to prevent infections in the future. We can identify common causes of outbreaks. And what I would advocate that countries do, or if you're in a region or a province, is to keep a list of the outbreaks that have occurred and you've investigated, and identify what the actual causes of each of them are, because what you do find is that common things occur commonly, and that the key causes of foodborne disease that result in outbreaks will end up occurring repeatedly. And you can identify a problem with a certain food product or a way of preparing food. And then finally, foodborne diseases are preventable causes of disease. It's worth pointing out, though, that not all foodborne diseases manifest as outbreaks, and there's a bigger picture burden. So we know that the global burden of foodborne disease is extensive, and this is some work that was done by the World Health Organization estimating global burden in 2010. And you can see that there are approximately 600 million episodes of foodborne illness each year, with 420,000 deaths. The majority of those are diarrheal in nature, but there's also other sources such as invasive infections, helminthic infections, and chemicals. It's worth noting here that the number of illnesses and deaths from chemicals is vastly underrepresented, and that is partly because they're very poorly investigated or understood. WHO are currently revising these estimates. Next. So the basic recipe, what does it look like for a foodborne disease outbreak investigation? In terms of the context, what we're interested in is situational understanding. If you look at a good recipe book, they're always going to tell you a little bit of history about the recipe and where it came from and what you serve it with. And it's the same with foodborne disease outbreaks. When we have an outbreak we suspect is due to food, we need to go and understand the context. What are the circumstances surrounding food preparation, transport, consumption? Then the ingredients. The ingredients of a foodborne disease outbreak investigation are the minimum requirements that we need. And I'm going to go through a diagram on that. And then finally, the methods. How do we go about investigating an outbreak that's suspected foodborne illness? Looking at hypothesis generation, choice of study design, investigation, and reporting findings. Okay, next. So when we think about the situation or the context of food, we may have an outbreak of, it might present as gastroenteritis or something similar, and we need to understand what is the connection here. An outbreak is more cases than we'd normally expect in a given time or place, and that might be a cluster of cases of gastroenteritis. And what is connecting them all? Is it a meal in a home? Next. Or is it a meal at a funeral? Next. Or is it a meal at a restaurant? And if we were in Taiwan, we would probably be eating some delicious food like this. And no, it didn't cause an outbreak, but I just put it there for illustrative purposes. Next. Or it could have been prepared at a market. So the people who are ill could all be connected by one of these types of settings. And it's also important to note too that for many foodborne illnesses, they may have other routes of transmission, and it's not always food. It may be water, maybe contact with other infected people, or contact with other infected animals. So it's really important as an investigator to keep an open mind at the beginning of an investigation and do good hypothesis generation. Next. So in all outbreak investigations of foodborne illness, there are really three main streams of evidence, and they're the key ingredients. So we need to gather evidence around laboratory testing. So that might be testing of foods, waters, clinical specimens. We need to identify the etiology responsible for the outbreak. We may test food and water samples collected during environmental sampling. And ideally, we would do some advanced testing of those samples to try and identify relatedness of strains that have been isolated from different sources. The next source of evidence or ingredient is epidemiology. We need, and this is where you come in as the FETP fellow, we need to give quick and clean epidemiology. It's not quick and dirty, but when it comes to outbreaks, we need to be rapid. Everyone who's been involved in COVID-19 for the last 20 months realizes that you have to give results today. The minister cannot wait. And it's the same with foodborne disease outbreak investigations. We need a good descriptive analysis of the cases that have occurred. We need to select the right study design to try and answer the question of what food or what ingredient caused the illness. And then finally, we need really good rapid reporting. So there needs to be rapid reporting to stakeholders and then rapid reporting to those who have a need to know. And then finally, food safety. Trace all the foods, collect the samples, and then identify and fix problems. And if we have at least two of those, we can achieve optimal management of the outbreak. Ideally, we have all three, but if you only have one of them, we really are not going to manage the outbreak properly. Next. So when we talk about hypothesis generation, and this is a key step at the beginning of pretty much every outbreak. So when we first get a cluster of cases reported, we need to 10 interviews, identifying demographics and clinical manifestations. We should collect an extensive history, not just focusing on food, but also water, animals, other infected people. We need to collect clinical specimens on at least 5 to 10 people. We also then would conduct an analysis showing the descriptive epidemiology of cases. And then we'd finally identify the agent and pathogen through the testing of those specimens we've collected. So we need to follow guidelines. I often see investigations where people found one positive in 10 samples, and it might be an organism which is commonly carried by people, and therefore doesn't really make it the cause of the outbreak. So we really need to follow good guidance. And I'll give you some references at the end that you can go to. The key reasons for identifying the agent is that it directs the type of investigation we might conduct. So if we wanted to, if it was an emerging pathogen, we would do a much more robust investigation than if it was something that was common in that setting. It also guides our control measures. So it gives us an understanding of where the contamination might be coming from and how we might go about stopping it. So if we find salmonella and it's related to consumption of raw eggs, we know that we have to do health promotion and stop people eating raw eggs. And all of this leads us to our hypothesis about what caused the outbreak. Next. And this is just a reminder to me to make sure you're collecting specimens and capturing them and sending them to a central laboratory, not just letting them go to any laboratory, one that you look after them, and ideally do the most advanced testing you possibly can on them. So next. I'm going to go through two examples, and this is the first. This is an outbreak of Salmonella enteritidis that occurred in Shenzhen, China, in 2018. And they had 10 cases reported to 2 hospitals, the local provincial health department, 10 people with gastroenteritis, all of whom lived at the local university campus. None of them knew each other. The investigation involved food histories, clinical manifestation, capturing clinical manifestations and demographic details. And what they identified was that all of them had used a online food ordering system on the same day, and they'd all ordered a consistent dish, which was chicken leg with rice. So the team went and they conducted testing of the restaurant. They conducted testing of the humans and the foods and the staff and the environment in the restaurant. I don't generally recommend testing staff in foodborne disease outbreak investigations. It's too difficult to know whether they acquired it from the food, and certainly it's probably less likely they contaminated the food. And what you can see on the left here is that there was the outbreak strains are all up the top, and there's a range of other sporadic cases of Salmonella enteritidis, and you can see there's a lot of diversity, but all of the outbreak strains are almost identical here, and there's included some chicken leg isolates in there as well. So you can see very clear association with the food. So here we have 2 ingredients, which is the food testing and environmental, and we also have descriptive epidemiology, which is excellent. Next. So the next step is we need to decide the study design that's going to answer our question. And in that previous one, too few cases, can't really do a study apart from descriptive. And that's what I've shown here on the right-hand side. If we can identify the entire group, that's great, that's easy. We can do a cohort study. We can sample the list of people if it's a large group. And if we can't identify people, then we go to a case control study. So we have an unclear denominator. We can use binomial probability if food preferences are known as well. Next, please. So this is an outbreak of listeria that occurred in 2017. And I'll just spend a little bit of time talking about this because you can see here, this has occurred over a 9-month period here. There were 937 cases. And 27% of these cases died. Listeria causes meningitis and septicemia. The key thing here is there were early signals of this outbreak. It wasn't required to be reported to the health department, but the hospitals noted it in July, August of 2017. And then there was a gastroenteritis outbreak at a nursery in early 2018, where they actually tested stools and identified the same strain as these other cases of invasive listeria. So they identified that there was a common food, and they conducted a case control study. And a couple of months later, in March, you can see they recalled products from a company. Next, please. And here you can see a picture of the meat. So it's a very large and serious outbreak of listeria here. These outbreaks probably go on all the time, but we miss them because we're not testing for listeria and testing in blood cultures. And this eventually resulted in a recall of these meat products. It's the middle one there, the pink one called polony. And it was exported to 15 other African countries. South Africa now has surveillance for listeria, and it was a watershed outbreak which changed their practices. Next, please. So just a couple of points. Another ingredient, of course, is the investigation team, and obviously that's a key part of initial formation of an investigation team is critical, and it's often multidisciplinary for foodborne disease outbreaks. It may involve departments of agriculture, local food safety authorities, departments of epidemiology, which can create tensions. Next. And again, as I mentioned, reporting is critically important here. It is something that we need to do rapidly. Next. Next. So my do's and don'ts, and I'm finishing up. Firstly, we need to form a team rapidly. We need to conduct early interviews to work out what is our hypothesis and collect specimens. I would advocate that countries need to develop outbreak database to develop a list of outbreaks and what was the implicated foods so they can look for common patterns. And I would say as an FETP fellow, you should investigate with passion. And it's true with cooking or with investigating. If you do it with a passion, it will lead you down the right path. Some of my don'ts: don't delay investigation. You need to do it today, not next week. Don't assume food is the cause. Sometimes it can be exposure to animals. Don't interview only sick people. And I think that is a flaw. We need to think about who's our comparison group. How are we making decisions about what is the cause here? And don't rely on food testing alone. We need those 3 forms of evidence: laboratory, epidemiology, food safety. And I've put up a big question mark here because I'd like you to think about what are your do's and don'ts. Next. So in conclusion, I think there are many lessons from COVID-19 which we need to incorporate into our foodborne disease investigations. Everything from using electronic records to identify where people have been and what they've eaten and what they've done. And here we could change that to what they've eaten. Early investigation is critical. Collecting specimens, using advanced testing like whole genome sequencing where possible. Follow guidelines, rapidly communicate, and think of new ways of doing things. And I hope to see more of that in the region. And then finally, don't give up. It's persistence which pays off. And sometimes you need to entertain a new hypothesis for the cause of the outbreak. Next. Here are some resources, and hopefully we'll be able to make these slides available after the conference. Next. Thanks. And next slide. And I just would like to leave you with the comment that if you have a foodborne disease outbreak, you're welcome to contact me, and I'm very happy to work through things with people. So very happy to hear of outbreaks that are difficult, because it is something worth fixing. Next slide. Thanks. I'll hand over to Dr. Chen.
Okay, thank you, Dr. Kirk. Is there any question?
Right, so I think there's a one question from the Q&A.
Oh, okay.
Yes, I can answer that, Dr. Cheng. Yes, I think I totally agree about the problem of
Martyn, sorry.
Yeah.
You may want to read out the question first, because I'm not sure if everybody can see the Q&A questions there. Thanks.
So there's a question here from Dr. Yudi Wibowo. Outbreaks of food poisoning are still common in several countries, including Indonesia. I think it's related to environmental sanitation, personal hygiene, and how to manage food in Indonesia. Poisoning occurs sporadically due to mixed drinks of alcohol or methanol and others in Indonesia. It's called oplosan and often causes death. Regarding this, please share your experience in Taiwan. There may be a solution to prevent death from oplosan poisoning. Thank you. I think rice wine poisoning is very common in the region, and it results in disastrous outbreaks of fatal illness. And I mean, the best solution is regulating and stopping that, but people make rice wine and it causes big outbreaks. I don't have any solutions, but I definitely agree it is a big problem in the region. I think hasn't been given enough attention. I don't know whether Dr. Cheng has any comments on that.
I have no comment because I didn't know this in Taiwan. Maybe someone, do you have any? Okay.
Yes, so rice wine problems or these, counterfeit alcohols used to be a much bigger problem in the past, but with regulation and much more knowledge, this has become much rare. So we haven't actually seen any of the these fake alcohol or counterfeit alcohol poisoning for a number of years. Over.
I think they have a problem, a question from Egos UD Paranormal from India. Thank you for share about outbreak investigation in a country or region with limited laboratory capacity and frontline epidemiology resources. The main problem is collecting specimens for an outbreak and carrying out investigations that are not standardized, so that the result of investigations tend to be difficult to use as material for taking control policy in the future. What can be done to overcome the problem or lack of capacity of epidemiological frontline resources? This question is for you.
Thank you. Yes, I think it is a huge issue to standardize investigations, and particularly in a big country with many provinces like Indonesia. I think the main ways that I would tackle this would firstly be to train more field epidemiology training program fellows, and they should in turn train other people. What we have done in other countries is run training workshops for investigating foodborne disease, and you can do things even where your laboratories aren't very sophisticated, and that is often one of the first things that is lacking. So I think training is important, and the other thing is developing standard tools for investigating. So I would advocate that countries develop standardized questionnaires and a standardized protocol for investigating gastroenteritis and foodborne disease outbreaks.
Okay, thank you. Because of time limit, I think still have some questions, but I think these questions maybe Dr. Kirk can answer online. Then
yes.
Thank you, Dr. Kirk. Thank you, Dr. Kirk.
Thank you, Dr. Chang. Thank you, everyone.
This third topic was the speaker is Dr. Chen-Chih Chen. He is an associate professor, Institute of Wildlife Conservation, College of Veterinary Medicine, Pingtung University of Sciences and Technology, Taiwan. Dr. Chen is a veterinarian, and he completed his PhD program in the Department of Large Animal Clinical Science, University of Saskatchewan, Canada. And later on, he came back to Taiwan, had been assistant postdoctoral fellow, assistant professor, and then promoted to associate professor in the Institute of Wildlife Conservation, Pingtung University of Science and Technology. Today, he is going to share a topic on integrating the knowledge of epidemiology and wildlife ecology for understanding the ecology of wildlife origin pathogens. Let's welcome Dr. Chen. Dr. Chen, please.
Thanks, Dr. Zhang, for your introduction. And it's my pleasure to be here to share some of our study in the past few years, and especially focus on wildlife disease and wildlife zoonotic disease. And can we go to next slide? Dr. Chen just mentioned I'm a veterinarian, and please allow me to introduce more about my background. I got my I have an undergraduate degree of veterinary college, and I got veterinary license. And after that, I do some practice in wildlife clinical medicine. And then I got a master's degree on wildlife ecology. After that, I joined some wildlife field survey bureaus, and I worked on wildlife ecology for a few years, and then I got my PhD degree focused on epidemiology. So for my background, I'm a veterinary clinical veterinarian, and also ecologist, and also epidemiologist. So in past probably 10 years of my research, we mainly integrate this background and focus on the field study for wildlife disease or wildlife zoonotic disease. So in today's topic, please next slide. Just click. Sorry. Okay, here. So in today's topic, I would like to discuss about the wildlife origin zoonotic pathogens, and how these pathogens transmit to domestic animals, and or these pathogens transmit to human populations, and especially focus on contact and the density issues. We use different kind of technology or different kind of method to evaluate or to estimate the contact of wildlife species, and of the contact between wildlife species and the domestic animal, or the contact between wildlife and human populations, and also estimate the density in different wildlife population or in the domestic animal population. Next slide, please. I think most of the participants in this conference are familiar with emerging infectious disease, and you might also know that most of emerging infectious disease are originally from wildlife. And the reason that we pay attention of wildlife original zoonotic pathogens in the field is that because most of people recognize that wildlife are the main reservoir of many pathogens, but why and where and when the disease or the pathogen can transmit to domestic animal or to human population is still remain a big issue in the research field, or both in the management or disease control agency. And we also usually ignore that the disease occurrence or disease reemergence is mostly due to the human activity that induce the habitat loss, or like climate change, or loss of biodiversity. So next slide, please. So when we focus on the disease transmission, we usually consider about the contact between individuals, although in different other different disease may have different contact systems. When we look at the primary drivers of emerging infectious disease, there are several main drivers, but the primary the main driver is the land use change. That's due to the land use change also change the contact between wildlife and domestic animal or the wildlife and human populations. And also we also know that the density, the agricultural industrial change also changed the density of farm animals, like in pig farm, poultry farms, which have very, very high density. And when the pathogen transmit or spill over into the domestic animal, which usually it can be transmitted very quickly, and possibly also influence or effect on the violence evolution. Next slide, please. This is an example of the wildlife original these pathogens transmitted to human. It's Nipah virus. Originally, Nipah virus, we know it's the main reservoir of Nipah virus is in the Pteropus beds population. But due to the habitat loss or habitat destruction or fragmentation, these bats, they migrate to some other available habitat, like usually like in agriculture area. In that area, they have many fruit trees or some domestic animal farms. And so these bats, they eat fruit in this area and dwelling in a farm. And at this time, they transmit the Nipah virus to domestic pig. And in this area, in the farm, they have very high density of pig population, and also pig is very good amplifier of Nipah virus. And so named because very high viral titer, it's easy to transmit to humans who are related to the pig farms. Next slide, please. So the contact and the density can influence the transmission of virus from wildlife to domestic animal or wildlife to humans. But the density can also influence the evolution of violence. This is very example of to figure if the high valence pathogen, it can usually can induce very severe clinical signs. And this kind of situation, it decreases contact between individuals. But if a pathogen is less violent, then the infected individual might have ability to walk around and transmit to other individuals. And this is usually time. But when the infection is in the high density population, then although high valence can induce very severe clinical signs, but the virus can also be transmitted very easily. Just click the slide, please. So the host density can influence the evolution of violence of different pathogens. But this is the common knowledge of epidemiology. But for wildlife, when we need to estimate or we need to evaluate the transmission between susceptible individual or infected individual, we need to know their density, but we also need to know their contact between individuals, or we need to surveillance to identify the contact between reservoir host and the spillover host. So for wildlife disease surveillance, we need to be available to monitor the contact between infectious or susceptible individual or reservoir host and spillover host. And we also need to be available to monitor the population dynamic, like population abundance. Next slide, please. However, for wildlife, it's not so easy to identify or to monitor the contact between different species or different individuals. So we need some tool, we need some knowledge to monitor the contact between the species or the individuals. So this is the example of our study. We used a GPS telemetry tracking system to identify the contact between the wild dog and the domestic dogs. So as we know, in the wild dog population, they have very high avian influenza diversity. Although most of the avian influenza are low pathogenic, but through the contact to the domestic dog or poultry, the avian influenza transmitted to the domestic dog or poultry. It is relatively easy to transfer to high density. It's relatively easy to change to high pathogenic avian influenza virus because it's in very high density situations. So next slide, please. In this work, this is the work we published in last year. We identified the virus strain from domestic poultry, dog farm, and from the wild dogs. And we found that, please click the slide, please. Yes. In this work, previous slide. In this work, we found that the high pathogenic avian influenza virus, they have some gene like MP gene originally from some wild dog. And these wild dogs virus strain are originally low pathogenic one, but they pass the gene to the virus endemic in the chicken farm or dog farm in Taiwan. And you can see the virus H5N2 in this figure here. They share they have very high similarity of MP gene between the virus originally from wild dogs, like northern pintail. Next slide, please. In the field, we also observed that in Taiwan, there are many dog farms, and these dog farms, the management methods are open to the sky, and the wild birds can easily go into these dog farms, especially for wild dogs. Their habitats are quite nearby the dog farm site, so they can go easily into the dog farm and dwelling in the dog farm because they are more abundant of food and shelters. So they on the right figure, in the right pictures, you can see that in circle area there are two wild dogs, and they mixed together with the domestic dog. Next slide, please. So in this study, our idea is that if we can identify where these wild dogs like to go, and when they migrate during the winter time into Taiwan, where they like to go, and is that possible these areas have high risk to transmit avian influenza virus to different domestic animals like dog or chickens, or even possible transmit to human population. So the first work is to identify their location and the name to evaluate, use different wildlife ecology model to evaluate their habitat selection. And then we can compare their habitat selection and with the current situation, current distribution of avian influenza virus in the poultry farms. Next slide, please. So we trap wild dog in the wetland habitat, and then we set up a GPS tracking system, and then the location of the dog, they continue to transmit the data back to us, and then we can use the GPS location to work on the habitat selection models, go through like a spatial scale of movement, and select a variable habitat type and the different kind of environmental factors based on the GIS system. Next slide, please. So this is our result, and we found that some critical factors are important for the habitat selection of the wild duck in Taiwan, and especially for the dog farms. We found that the wild duck like to choose the habitat very close to duck farms. And I just mentioned, we observed many cases that with these wild duck, they go into the duck farm and mix together with domestic dogs. And in our model also show that duck farm is a very important factor that can influence their habitat selection, and they prefer to choose the habitat close to the duck farms. So in this kind of event, it's very high chance that the avian influenza virus carried by the wild duck can be easily transmitted to domestic dog or chickens. And then in this kind in a high population density situation, it's possible that the virus, low pathogenic avian influenza virus, can evolve into the high pathogenic avian influenza virus. Next slide, please. And these are other factors that have a significant association with their habitat selection of wild duck. Next slide, please. And also, we based on the statistic models, and we create figures show their habitat selection. This is the middle on the middle of the figures, and the red colors means that it's habitat that dogs wild dogs prefer to stay during the winter times. And on the left figure, it shows the high pathogenic avian influenza virus outbreak in different areas. And the red color also red color shows that this area have a very high number of cases with high pathogenic avian influenza virus. And the right figure shows the distribution of poultry farms. And based on comparison of these figures, we can find that in some area with high density of poultry farm, and also the high preference of habitat selection, then you will have a very high cases of pathogenic avian influenza outbreak. And but if in some area with high poultry farm but not prefer habitat, then you have very low pathogenic avian influenza outbreak. We're still working on this part. We try to, based on the statistic model, try to evaluate the association between habitat selection and also avian influenza outbreak. And we also try to use the data to validate the outbreak in the following years, the outbreak of avian influenza virus in the following years. Thank you. Next slide, please. And the next case is a rabbit in Berry Beijing in Taiwan. We have outbreak, a big outbreak of rabbit in Berry Beijing population. Is a wild cannibal species belong to the Masteridae in Taiwan in 2013. And the next slide, please. So you can just click the slide and show the spread of the rabbit virus in Taiwan. So you can see that the original outbreak of rabies virus is in the central area of Taiwan, and then it gradually spread to south area and east area. And after the outbreak, we do a very long-term observance. And in our research project, our research team mainly focus on how the rabies virus influence on the rabbit fairy badger population, and how fairy badger population, the change of fairy badger population influence the cases or the transmission of rabies virus in the fairy badger, the main reservoir of rabies in virus. Next slide, please. So we trapped the individual, and we put the telemetry tracking system on this fairy badger, and we estimated the density of fairy badger before and after the outbreak of rabies in each specific sampling area. And then we try to estimate the R naught and the density ratio. We hope that we can understand the density ratio of rabbits in fairy badger population. Next slide, please. So it is a procedure of density estimations. So we first do the animal tracking, and then we say that the animal and make a mark on the animals, and then we release into the original habitat, their original habitat, and then we install the auto the auto camera in the field, and so the camera can detect the animal and take pictures in the field, and you can see the picture on the left side below lower one, and you can see that this is a fairy badger and with a mark on the neck, and then we can identify specific individuals. So through this process, we can estimate their density, and then we can also tracking their activity, see if they are still survive in the endemic area or they died due to the rabies virus. And through this process, we can estimate density and also estimate the transmission or not. Next slide, please. So this density estimation goes through this process and also adopt different type of models. We like a capture recapture model, spatial capture recapture model, or mark recite model. We can estimate the density and see before and after the outbreak of rabies. Also, we continuously do a surveillance and see on what kind of density threshold or what kind of density situation the disease occurred or disease can transmit to different individuals. And also estimate density have another very important part is that because during the outbreak and also after the outbreak, our government tried to management or control the spread of rabies in Taiwan. And the original target is use of oral rabies vaccines in the field for these fairy badgers. And but we need to know the density, the distribution of density in different areas of fairy badger. And so we need to precisely in what kind of habitat or what kind of forest type the density of fairy badger or the different the different density of fairy badger in different habitats. So you can see the figure on the right side. This is a map show the density in our sampling area. So goes through this density estimation model, we can estimate, although it's in the same sampling area, but with different habitat type, you have density situations. Next slide, please. So based on this density estimation, we can estimate the rabies vaccine deployment programs, and to estimate in each specific area how many vaccine we need to deploy in the habitat of fairy badger. And this the figure on the right side is the plan that we try to estimate the vaccine we need to deploy in the habitat of fairy badger. Next slide, please. So for example, like in the East Taiwan, there is a major river, and we consider that this river is a important natural barrier that prevent the spread of rabies from south to north. So if we want to prevent or management the further transmission, expansion of rabies in East Taiwan, we try to deploy the oral vaccines in the south bank of these rivers. So these are different habitat types include the agriculture area, forest area, traffic, water body, building name. Because Flypager is a forest dwelling species, so the only thing we need is to deploy in the forest habitat, and the total area we need, and also the total amount of overall rabies vaccine we need. Okay, next slide, please. So at the end, because we put a lot of effort on trying to surveillance or to screen the pathogen different microbes in the wildlife populations, and it's very important work. But however, if we want to prevent or identify the risk of the transmission or spillover of this pathogen from wildlife population to domestic animal or to human population, it's very important to identify where is the contact and to identify some other environmental factors which influence the contact or influence the densities. And for wildlife disease surveillance, we need a lot of knowledge and a lot of tool skill from the wildlife ecology part. So we can adopt this tool or this knowledge and to combine with epidemiology concept, and then to evaluate the disease transmission of wildlife disease transmission from wildlife population to human population. And I think this is all my talk. Thanks everyone, and also thanks to Dr. Chen, the chairperson. Thank you.
Thank you, Dr. Chen, for the sharing of the avian influenza and animal rabies in the Flypager in Taiwan. I think they have one question. Is there any question? No, they have. This is. There's one from audience. Say, thank you, Dr. Chen, for a very interesting presentation. Is there any questions? I think we are, okay, one question. I think we have one question from Malaysia, Dr. Chu Wei Li. Good work, Dr. Chen, and to your team, challenging indeed. On an average, how long the tracking device is fixed on the wildlife? Okay,
it depends on different situation, like how long of the study, what's your purpose of your study, and also very important part is the size of animals. And because if you have a large size of animal, you can have a large tracking device, and then you can have large battery which can last longer, and on the different kind of species of wildlife. But as I mentioned, it depends on different purpose. If we just need 1 year of study, then usually it's enough. Usually even for very small, like a 10 gram or 15 grams device, it can last for something like 1 year. So usually it can longer than 1 year, but depend on your purpose.
We have a short question. I think that the last one is, can you tell what are the most sources of the infection from?
I think the same. It also depends on different pathogens and also their reservoirs, because different wildlife species have their own different habitats. But for my opinion, I would say that in agriculture areas are higher risk of transmission of wildlife disease from their population to the domestic animal or to the humans, the agriculture area. Yeah.
Okay, I think the animal rabies is the origin is still a riddle in Taiwan. But you are
sorry.
I think the origin of the animal rabies in Taiwan.
Yeah.
What's your consideration on
the origin?
source? Yeah.
Of course, the origin will be the dog. from the dog, and the name is, we don't know why and how it transmits to very big population, and the population, the very big become the main reservoir of rabies in Taiwan at this moment. But we also worry that because the original strain is from dog, it is possible that it spill over back to the dog population. And in Taiwan, we have high density of stray dog in the agricultural area, in the rural area. So so our government now try to increase the coverage of the vaccination rate in domestic dog and the cat. But however, the stray dog part, it's very difficult to reach high coverage of rabies vaccines. So it's a risk there. And usually these stray dogs, they distributed in the rural area or agricultural area.
But
So,
up to now, we didn't have any single stray dog has positive rabies.
yes,
in
we
Taiwan.
do. We have one case in Taitung.
I think that is a very baby dog.
Yes, very.
I think that is a baby dog, about 4 months or 3 months old.
Yes,
Yeah,
Dr.
I
Chen,
think, yeah.
yes, Dr. Chen, you're right. And that dog is originally it infected because it fight with infected peribeger, and then the virus transmitted to the dog in the Aboriginal village. Aborigines village. So, and then it the symptom shows, then it was put in quarantine. Yeah.
Okay, thank you, Dr. Chen, for your interesting presentation. Thank you. Then
Thank you.
this section will be closed here. Okay, thank you.
Okay, thank you.